Alright, so now that we've got a basic overview of these large macromolecules, we're going to look at each one of the categories, all four of them, one by one, and we'll break this up into a vodcast for each one of them just to keep it kind of small and so that we don't overwhelm ourselves. Now as we go, you're going to see some repeating information, some repeating patterns, and that will help you kind of tie all this together. Concept 5.2 talks about carbohydrates, and it says carbohydrates serve as fuel and building material. So carbohydrates is one of those four categories of macromolecules that we're talking about, and carbohydrates include sugars and other polymers of sugars. We're going to find out sugars and starches and the like. These are all carbohydrates. So if you're thinking about your human diet, you might think of things like sugar and bread, pasta, things of that nature would be carbohydrates. The simplest carbohydrates we're going to find are some very basic simple sugars called monosaccharides. The mono means one or single. The saccharide is the scientific word for sugar. And then these monosaccharides can be put together in these long chains, and they can create larger molecules. We're going to find out those are called polysaccharides. So carbohydrate macromolecules are polysaccharides, and these again are long chains of repeating patterns, and they are polymers composed of many sugar building blocks all stuck together. So let's start out by looking at some of the sugars because they tend to be some of the more simple ones, and we'll start by looking at monosaccharides. So monosaccharides are the very simple, less complex molecules that we're talking about, and they usually have a molecular formula that is some multiple of CH2O. So for example, glucose, which is probably perhaps the most important and most common monosaccharide, is C6H12O6. So you can see that's a multiple of CH2O. So if you multiplied 6 times everything you see here, you would get C6H12O6. So this formula is a multiple of this basic kind of layout right here. Monosaccharides are classified by two different sort of chemical signatures, and it has to do with the location of its carbonyl group. And if you remember when we were talking about functional groups a chapter or so ago, the carbonyl group was one of those functional groups. And I've put it here on the page. If you'll remember, it's a carbon with a dual or a 2 covalent attachment to the oxygen. So this is a carbonyl group. It's one of those functional groups that makes up a part of many important biological molecules. And so a monosaccharide is classified based on where this functional group is located. And you've already seen some of these words actually previously in other chapters. If it's on the end, for example, I'll show you a picture in just a moment. If this carbonyl group occurs at the end of the molecule, we call it an aldose. And if it occurs somewhere in the middle of the chain, we call it a ketose. And the number of carbons in the carbon skeleton for these sugars generally ranges from 3 to 7. So these are pretty small, only 3 to 7 carbons long in the main part of the chain. That makes for what you're going to find out is a pretty small molecule compared to some of the big ones we're going to look at in just a few minutes. So for a picture, so here we have the structure and classification of monosaccharides. And if you'll look closely, these are called trioses because they're 3 carbon sugars. Very simple. They've got 3 carbons kind of as a backbone. And then they have H's and OH's hanging off the edges or sides. And both of these, if you notice, are the exact same formula. They're both C3H6O3. But one of them is considered an aldose because you'll notice it has its carbonyl group hanging off of the end here. And this one is considered a ketose because its carbonyl group is somewhere in the middle. The actual names of these two sugars are down here below, but the categories are right up here, aldose and ketose. If it's got the carbonyl group on the end of the molecule, it's an aldose. If it has the carbonyl group somewhere in the middle, it's called a ketose. And again, remember about a chapter or so ago, what did we call a molecule or two different molecules that have the same chemical formula, right here, the same chemical formula, but they have different layouts or different three-dimensional patterns to them? Well, if you'll think back, we called those isomers. So an isomer is 2 different versions of a molecule with the same chemical formula but with a different layout. And these are, of the isomers that we talked about, these two are structural isomers. In other words, they're layout is a little bit different, where you can see that their pattern or their shape, as far as like a line drawing, is a little bit different. So these are structural isomers. They have the exact same formula, but they're considered two separate molecules. And it's interesting, or it's odd in biology, how the layout of the molecule is sometimes, or in many ways, just as important as the actual chemical formula. The chemical formula tells you a lot. It tells you what's in there, but it doesn't necessarily tell you how that molecule is put together and how it's laid out in a three-dimensional way. So we were saying that most of these simple sugars, these monosaccharides, tend to be from 3 to 7 carbons long. And going back here, you can see that these are only 3 carbons long. They're some of the smallest ones. Here you have some larger ones. These are called pentoses because they're made up of 5, and pinto is 5. So these are 5 carbon sugars. They have a backbone here of 5 carbons, as you can see. This is ribose and ribulose, which are, again, structural isomers of each other. They have the same chemical formula, but they're laid out slightly differently. And this puts them into the 2 different categories. Again, the aldose has the carboxyl group, the carbonyl group hanging off of the end, and the ribulose or ribulose has the carbonyl group somewhere in the middle. One last example of some of these sugar molecules, the monosaccharides. Here we have an even slightly larger sugar. This is a 6-carbon sugar, and again, it's got a base or a spine of 6 carbons here and here. And these 2 categories are isomers of each other. They're structural isomers of each other. But on the aldose side here, where we have the carbonyl group that's on the end, we actually have even a greater number of isomers. We have 2 isomers of each other within the same aldose category here. So these are, if you look closely, they are cis-trans isomers. Sometimes, remember, we call them geometric isomers, cis-trans isomers. And that's because if you look right here where they have it in purple, you can see how in one case they are flipped, like they have the H and the OH bond on different sides. So this would be cis-trans isomers. And these 2 are glucose and galactose. They are cis-trans isomers of each other. And both of these, glucose or galactose, are structural isomers of this guy over here, fructose. So you see we have the 2 categories. We have the ketone or the ketose sugar because the carbonyl group is somewhere in the middle of the chain. The aldose or aldehyde sugars because the carbonyl group is on the end. But we can even have isomers of isomers, which kind of gets confusing. But these are all what we call monosaccharides. They are the fairly small, simple sugars. Up to this point, we've been drawing all these sugars kind of in a linear skeleton or a line drawing type form. If you go back and take a look, we're drawing everything kind of in straight, nice lines, showing that carbon backbone as a single, nice, straight line. But in reality, oftentimes when they're in a liquid solution, a water-based aqueous solution, many of these sugars form into the shape of rings. And so I'll show you a picture of that in just a moment, or a drawing model of that in just a moment. But in real life, oftentimes these are in kind of a ring shape as opposed to that straight line shape. Monosaccharides are important because they are the major fuel for living cells, and they're the raw materials for building molecules. So for example, that monosaccharide we talked about, glucose, it's probably the most important sugar, the most important small sugar monosaccharide in the entire history of life. It is central to the importance of the chemistry of life. It's the sugar that's created through photosynthesis. It's the sugar that human body cells and animal body cells use as a primary source of energy. So monosaccharides are quick energy, easily used by the body, easily broken down, and either used for energy or used for building blocks for other parts. Here's a picture or a drawing of the linear and the ring forms. So if we were going to make it simple, we'd probably just leave it in this ring format right here. But in reality, they tend to form these ring shapes that kind of would be drawn out in this way, this model on the right. And then there's other ways to kind of abbreviate it. You'll notice as you go throughout your science career, especially into college, that scientists are just like teenagers in texting. They tend to like to abbreviate things and either come up with ways to show or say something so that you don't have to use the entire name or give all the information. So this is oftentimes what you'll see. This is an abbreviated ring structure, and what they've left out is at each one of these junctures, if it doesn't specify, it's just supposed to be known that there's a carbon at that juncture. So like if you compare this one to this one, what you'll notice is they've left out all the carbon atoms that join at these junctures. So many times when you take upper level chemistry or upper level biochem, you'll notice these abbreviated ring structures to where they've left out certain information that just as a general rule, you know that's representative of carbon. So we started off with the smaller sugars, the more simple sugars, and those were called monosaccharides. But of course we can get larger. We can get up to what we call disaccharides, and a disaccharide is when two monosaccharides are joined together. That word di, or the prefix I should say di, means two. So a disaccharide is when two monosaccharides join together and link up. Like it would be like taking two different chains and linking both those chains together to form one single larger chain. And the way this happens is through what we call a dehydration reaction. Remember, a dehydration reaction is where basically you are producing or losing water kind of as a byproduct. And the covalent bond that links these two chains together is called a glycosidic linkage. So if we're going to make a disaccharide from two monosaccharides, it's going to be a dehydration reaction that joins them together. And then that linkage, right where they're joined, that's a covalent bond called a glycosidic linkage. And a great example of disaccharides are when you take two glucose molecules and link them together. You create a disaccharide called maltose. And maltose is an ingredient, for example, that's used in brewing beer. And then if you take a glucose and you link it with a fructose, you get perhaps what's the most commonly known sugar for humans, which is sucrose, which is what you put on your cereal or what you put on a grapefruit. It's what you put in your tea. That's basic table sugar. So basic table sugar, sucrose, is a disaccharide. It's a combination of two monosaccharides, glucose and fructose. Here we have the basic animation that would help you out and show how these two monosaccharides join together. So here, if you wanted to create sugar, table sugar, you would need two monosaccharides. You need a glucose and you need a fructose. And those two would join together with a glycosidic linkage, and that would create a disaccharide. And the disaccharide it would create would be basic table sugar, sucrose. So take a look at this animation. It may help you out, and right-click and hit play, or you could see it in the PDF form as well. Here we have a picture of the same thing. So again, here we have our glucose molecule. And then here we have two glucose joining together, and we have this H2O water produced, dehydration reaction. And this linkage here is called a glycosidic linkage. And what have we created? Well, we've created the disaccharide maltose. And then down here is the other one that we just looked at. Again, there's a glucose monosaccharide and a fructose monosaccharide. Join those two together, the dehydration reaction, that's called a glycosidic linkage. And we have created good old table sugar, sucrose, that we can add to our cereal. Alright, so we can go even larger, just like with a set of Lego bricks. You know, you can start out with a single brick, and then you can build a simple structure of 2 or 3 bricks, and then you can take multiple of those simple structures and add them together and build something even larger and more complex. Well, we do that with molecules as well. So now we're up to what we would call polysaccharides. So we had the monosaccharides, the disaccharides, and now we're just going to call the rest of them polysaccharides. Poly means many or much. So polysaccharides are long chains, polymers of sugars, and they have a very important role in biology because they're used for storage of energy, and they also have like a building block or a structural role. As energy storage, oftentimes they are kept as part of the body, and then when the body needs that extra energy, they are hydrolyzed to break them apart into smaller pieces and then used as energy. And as building materials, they can be used, we'll find out, to help build cell walls and help build other organ parts and cell parts that are for protection or for construction of the living creature itself. So whether or not they are building materials or storage materials, the structure and function of this long sugar chain, it's based on what sugar monomers, and the monomers are kind of like the individual links. It's based on which sugar monomers make up the chain, and it's also based on where these linkages are located on the chain, these glycosidic linkages. So that determines what the purpose is of the sugar and what the structure is of the sugar. What are the types of links that make up the chain, and where are these specific types of linkages called glycosidic linkages? Where are they located along the chain? So let's look at the storage first. So energy storage, living creatures don't have the benefit of plants for the most part. Plants can gain energy by using power energy from the sun, but most other living creatures don't have that benefit. They have to carry their energy stores around with them. And plants have to have an ability to store energy as well because the sun's not out 24 hours a day. So storage polysaccharides are what plants and animals use to maintain a certain level of energy within their living systems. And in plants, it's called starch. Starch is a storage polysaccharide for plants, and it consists entirely of long chains of glucose monomers stuck together. And plants store this starch within their chloroplasts and within other parts of their cells as little granules. And the simplest form of starch is something called amylose. So when you're eating bread or spaghetti or a vegetable, say like for example a potato, you're taking in a lot of that starch. And that potato is basically a storage system of energy for that plant. Amylose and all these other starches can be hydrolyzed, broken apart, and then the glucose monomers that make up this long chain can be released and used by the plant when necessary. Here's a microscope photograph of the chloroplast of a plant and these whitish sort of areas. These are starch granules. So this is the storage system within the plant, how it stores energy in these long chains of glucose molecules. It's like every glucose is a link in the chain, and when they're all chained together, they make what's called a big polysaccharide, and that's a good way to store energy for plants when they need it. So that kind of explains why plants don't get fat. You never see a plant that has love handles, so to speak. But we know the same's not true for animals. Animals can get fat, and we're going to talk about that actual lipids, those fats, a little bit later. But animals also have a way of using, it's kind of like the animal version of starch for short-term energy storage. And in animals, including humans, this is called glycogen. Glycogen is the storage polysaccharide in animals. So this is not fat. This is not like what you have in your lips and your rear end and on your hips. This is slightly different. It is a storage molecule, but it's a sugar molecule. It's a carbohydrate molecule called glycogen. Humans and other vertebrate animals that have a backbone, they all store this energy, glycogen, mainly in their liver and in their muscle cells. And here in this microscope picture, we have some muscle cells here. And you see kind of little darker areas that the arrows are pointing at. That's glycogen. And again, glycogen is basically the animal version of what starch is for plants. So when necessary, the body can break these chains apart and release the glucose and then use that glucose for energy. Now, glycogen in animals is really only short-term storage. For example, if you decided to skip a couple of meals, you're going to pretty quickly run out of glycogen. That's stored in your body, and you would go through all your reserves in about a day. So if you skip a day of meals or you go more than 24 or 48 hours without eating, your body is basically going to exhaust its supply of glycogen. It's strictly a short-term energy storage in animals. Starch in plants is long-term, but for humans, we're going to find we have another way for long-term. Here's a picture within an animal. You have some cells here. Shows some mitochondria within the cells, and then these little dark areas are granules or chunks of glycogen material. Here's the two of them together just to kind of show you. These two models represent two different versions of the starch, amylose and amylopectin. Both of these are fairly simple. Amylopectin is a little bit more complex. But then within the plant, this is how the plant stores energy. There's starch granules inside the chloroplasts, which are inside the plant. So this is a plant polysaccharide. Poly meaning many, so just a long chain is what we're talking about. Down here in the animal, glycogen, that's the form of it. And these dark little granules represent glycogen within the animal cell. Again, it's also energy storage, but in animals it's strictly short-term energy storage. So both of these are long chains of sugars called polysaccharides, and they're used as energy storage in living creatures. The other purpose for these polysaccharides, these long sugar chains, is structural. They actually make up the material that builds the cell or builds the organism itself. The polysaccharide that probably most people are aware of or know about is called cellulose. And in plants, it's especially important. Cellulose is the major component of the tough, strong cell wall of a plant. So if you look at the difference between a plant cell and an animal cell, one of those differences is the fact that plants have what we call a cell wall, and that's built of cellulose. And so it's much like starch. As a matter of fact, it is very similar to starch. It's a long polymer chain of glucose molecules all stuck together, but the difference is where are those specific linkages called glycosidic linkages. So if you compare starch and cellulose, they are a lot alike. They're both made up of long chains of glucose molecules, but where they differ is in the form of their linkage. Where those glycosidic linkages occur, that actually makes a huge difference. Cellulose wood is probably the most abundant organic compound on the entire planet. Plants produce about 100 billion tons of cellulose every year, and I know in the fall, when I'm raking up all those leaves, sometimes it seems like I'm raking up 10% of that out of my yard, but there is a lot of cellulose produced every year by plants. So the difference we're going to look at between these two, between starch, which is an energy storage polysaccharide, and cellulose, which is a structural building block saccharide, the difference is based on these linkages, these glycosidic linkages. And we're going to look at them. They're both in kind of a ring form, but they're called alpha and beta based on the two different linkages. Here we have an animation. If you want to take a look at it, right-click and hit play or look at it in the PDF. So this is going to show you an animation of polysaccharides, and we're going to look at a picture here of the difference between these two polysaccharides. So here we are. We have polysaccharide alpha and beta. One of them ends up being starch, and one of them ends up being what we call cellulose or wood, if you want to think about it in its everyday terminology. So if you look, they both have the exact same chemical formula. They're made up of a chain of glucose, or a chain of many glucose molecules stuck together. But look very carefully, there's a slight difference down here in where the bond is, right there. So compare these two in their chain form down here. If it's starch, the way that the glucose monomers are all linked together is with what we call the alpha glycosidic linkage. And notice that the OHs are all on the same side down here. And then look, here's cellulose. It's the same thing. It's made up of all these glycogens linked together. But notice how the OH is on opposite sides of each other. It kind of flips back and forth, up and down, up and down, up and down. And you wouldn't think this would make any difference. They have the same chemical formula. They're even the same sort of chain of glucose molecules. But this different ring structure, these differences in where the linkages are, actually makes a huge difference in the way that the molecule is shaped. So if it's a chain of glucose polymers and they have what we call the alpha glucose linkage, they're going to be what we call helical in shape. So if they have the alpha linkage, they're helical, kind of a spiral, and I'll show you a picture in just a moment. If they have the B or beta glucose linkage, they're in straight lines, and it makes a big difference. In straight structures, the H atoms on one strand can bond with the OH groups on another strand. So what that means is if they have this beta glucose linkage, they can actually form like lines that can lay on top of each other and bond to each other. So you have all these parallel cellulose molecules that can build up and stack on each other in a way that makes them extremely strong. So if they have the B linkage, it becomes what we call cellulose. And that makes a very strong, tough material that the plant can use to build its cell walls or to build the trunk of the tree or something like that. Always helps to see a picture. So let's take a quick look at a picture here. So again, this is that beta glucose linkage. If it's alpha, it's starch. So if it's an alpha linkage of glucose molecules, it's what we call starch. That's what the plants use for energy storage. That's what you're eating when you eat the spaghetti that came from the wheat or the potato that came from the root of the potato plants. But if it's this beta linkage, then they form nice straight lines, and they can be stacked on top of each other, and they can bond between these stacks. And that forms these cellulose fibers called microfibrils. And here we're zooming further and further out. Here's the molecules stacked on top of each other and connected. They can form these microfibrils. And then the microfibrils can form cell walls, like you see here. And those strong cell walls make up the structure of the leaf or the structure of the trunk of the tree. So the only difference between starch and cellulose is the type of linkage. If it has an alpha linkage, they're going to be helical, and the monomers, the long chains, and the polymers, they end up being helical in shape, and they can't stack up and join together, and so they're not quite as strong. But if they have this beta linkage, the way they lay straight and link together can form this very strong material that the plants can build itself out of, build its leaves and its trunk and that type of thing. And it makes a difference in what other animals can and cannot eat. So you have to have a different enzyme that can digest these two different forms. And the enzymes that can break apart starch can't break apart the cellulose, which is kind of odd because it's the same material. It's got the same chemical formula. But it takes certain enzymes to break apart those linkages. And the one enzyme that can break apart the starch cannot break apart the cellulose. So for example, humans. Can a human eat a potato and digest that potato? We sure can. That potato is made from starch, and that would be the alpha linkages of these glucose monomers. But can we eat wood? Could you go to Lowe's and buy a 2x4 and have that for dinner? Well, the answer is no. Our bodies have, inside our digestive system, the enzymes to break apart the potato, to break apart the starch, but our bodies do not have the enzymes to hydrolyze those beta linkages. So we can't digest cellulose. If we eat cellulose, whether it's a celery stick or in a salad, or if you were just to go to Lowe's and buy you a 2x4 for dinner, it's not going to do much good for you. It's going to pass through our digestive system as fiber, insoluble fiber that we can't digest, and it's going to end up primarily in the toilet. So humans can't live off of leaves and grass and 2x4s because we don't have the enzyme to break that beta linkage. There are some creatures that do. There are some microbes, tiny little bacteria and other creatures that do have the enzyme necessary to digest cellulose. And guess where a lot of these little creatures live? They live in the stomachs, in the digestive system of other creatures. So for example, a cow can eat hay, a cow can eat grass. Well, the cow has inside its stomach billions of these bacteria that can produce an enzyme to break down that grass and that hay. Same thing with termites. The termites themselves can't eat and digest the wood, but inside the stomach of termites are millions of microbes that can break that wood down, break these linkages in the cellulose. And they have what's called a symbiotic relationship. The bacteria are getting a nice, good place to live and breed and reproduce. And the flip side of that is the cows and the termites are able to get a food source and able to get that energy out of the grass and hay. Even some fungus, some fungi, some mushrooms and other fungus have the ability to break down cellulose, and they do have this enzyme. So when you see, if you go hiking in the woods and you see an old rotting log or an old rotting tree, and you see mushrooms growing out of it, those mushrooms have the ability to produce an enzyme that can break that wood down, break that cellulose down, and they can get energy from that. So they're living off of the decaying body, you would call it, of the tree. Some scientists even think that at one time, maybe humans did have the ability to digest this cellulose. That's what some scientists think our appendix may have been for. Our appendix is a little pouch that hangs off our large intestine, and some scientists think that perhaps it was larger in the past and it contained some of these bacteria that could help us digest cellulose, like leaves and grass and that type of thing. The reason they think that is because that's the way it is in a lot of other animals. In horses, for example, horses have a part of their digestive system that looks a lot like our appendix. And in the digestive system of the horse, inside that part, there are these microbes that produce these enzymes that break apart the cellulose. Just to give you one more example of a structural polysaccharide, a polysaccharide that's used to basically build the living creature, and that would be in the exoskeleton of arthropods. Arthropods are insects, spiders, crustaceans like crabs and lobsters. They have a polysaccharide called chitin. It looks CH, but it's pronounced like K, chitin. Chitin is what makes up the hard shell of an insect or the hard shell of a lobster, and it's kind of like their skeletal system. They have an exoskeleton. They don't have bones inside their body. They have this hard chitin shell to protect the soft insides. And chitin is also what provides for the structural support of a lot of mushrooms and fungi. So the reason the mushroom can grow up and stand up against gravity is because a lot of its cells use this polysaccharide called chitin as a structural polysaccharide to build its kind of a cell walls and material to stand upright. So here in this last picture you see one of these bugs that's emerging from his shell. That shell, as they molt and grow larger, they have to grow a new shell because that shell is basically that hard chitin. And as they grow, they have to shed that shell, and their body has basically built a new shell that's soft at first, and it hardens with contact with the air. So that shell, if you pick one of these up, of a locust, that's made almost entirely of chitin. And chitin is a long chain, a polysaccharide of these individual links called monomers. Another good thing, another cool thing, I guess, a usage of chitin is, believe it or not, some scientists or doctors in surgeries, they use chitin as a material to make surgical threads. So if you were to ever go into the hospital for a surgery where you had to be opened up and then sewn back together, the thread that they use to sew you back together, especially the thread that might be used inside internally, might be composed of chitin. It's very strong, very flexible, but can be basically dissolved or decomposed by the body over time. So if they were to sew you together on the inside, and they don't want to have to go back in there to take the threads out, they might use chitin-based thread that would decompose and be broken down by your body over time. We're going to stop right there. So these are all sugars. So the first category is saccharides or sugars. And so if we want to do a quick review, see, we'll jump right back to the front for just a second. All of these are carbohydrates. That's our main category, carbohydrates, and they're either going to be used as fuel. So a lot of these sugars, these mono and polysaccharides are either used for fuel, and that would be, for example, starch in plants or glycogen in animals, or they're used as building materials. And the building materials would be like the chitin or the cellulose that we talked about. They're all long chains of molecules. Monosaccharides are the simple small ones, and then we can have disaccharides, and then we get up to what we call polysaccharides, which are the very long polymers of many, many blocks of these sugar molecules. They're joined together by glycosidic linkages, and those linkages play a big part in what type of material they're used for. So, for example, with a long chain of glucose molecules, that could be starch or it could be cellulose. And what determines that is where and how those linkages occur. So we're going to stop right there. This was carbohydrates, which are sugars, and other polysaccharides. We'll stop, take a break. We'll come back with fats and lipids.